Hi, and welcome to this podcast on epigenetics. This will be our first podcast that we will do on emotional equilibrium, and I'm very excited about it. You are speaking to or listening to Natanya, and I will be your host for this epigenetics small course that we will be doing. And I'm very excited to start it off with you. We will do a few short sessions and not try and overcomplicate things in all at once. So we will be starting off with welcoming everyone to this session. Welcome. Please note that I'm a quantum soul and quantum life consultant. I'm not a medically trained doctor, psychologist, or psychiatrist. Should you display any symptoms that are worrying to you, your first priority is to contact a medically trained professional to assist you. So when we think about epigenetics, we always wonder, what is epigenetics? What does it mean? And I'm going to spend this course explaining to you what is epigenetics. What does it mean? How does it work? How do you get an epigenetic marker? How do you get rid of an epigenetic marker? What is an epigenetic marker? Okay, and how does that fit into epigenetics? So let's get started. So today we are going to look at what is epigenetic markers. We need to first understand what is an epigenetic marker. So epi, as you can see here, means on top. Genetic means your genes, and a marker is an indication of position or place. So on top of our genes, we have little things that in that place can switch on and off. Okay, so let's look at what does that mean. Okay, so to oversimplify this, on top of your genome sequence, so on top of your genes, there are these little markers that turns on and off, like light switches. And in order for us to understand how these light switches will affect our lives, we first need to understand what is the difference between DNA and genes. So if I ask you, like, why is it not a DNA marker? Why is it a gene marker? Why is it on top of the genes? Why don't we say it's on top of the DNA? We first need to understand the difference between DNA and genes. So when we think about ourselves and our bodies, every single part of you is made up of cells. So you have the cell, and inside of the cell, you have the structure that we call the nucleus. That is the main part of the cell. It's like the brain of the cell. And inside of the nucleus, we find our genetic information in the forms of DNA. Okay, so our DNA is basically caught up in these small things that we call chromosomes. Now, as humans, we have 23 pairs of chromosomes, meaning we have 46 in total. So you can see they are in pairs. And what happens if you think about DNA is it's the inherited biological information. So it's all the information you inherit, half from your mother and the other half from your father. The genes is a section of the DNA that has a specific function. It tells the body what to do. It's a section of the DNA that tells the body what to do. So to even more oversimplify this, let's quickly think about it like this. And I just want to move the video so that you can see this. So first of all, you need to think of the nucleus, which is inside of the cell, as the basket. Okay, and inside of the basket, you have yarn or wool rolled up in a bundle. So each one of these bundles of yarn that's rolled up will be your DNA that's rolled up into a chromosome. So each one of these will, and this is very oversimplified, I know that. So you have the basket, which is the nucleus, and the rolled up DNA that forms a chromosome. Okay, so let's go back. Sorry, I just want to move this back. Let's go back to this picture. So basically, if you think of your chromosomes as DNA that's rolled up many times to form one ball. Okay, so that will be the chromosome. If you roll that yarn or that ball of wool out and in length, what we will do then is we will have the DNA. And so the DNA is what we call it this DNA helix, but it looks like a ladder. And so the entire ladder, the whole thing, the whole string, the whole yarn, if you roll it out, that is considered, all of it, the DNA. Now, on the DNA, a small section of the DNA, like we see here, they will say gene. A small section of each part of the DNA will be considered a functional piece, meaning that these few steps, right, that is in the gene, that few steps will have a function, whether it be eye color or hair texture, or whether it be skin color. It will be the density of your nails. It will be the texture of your skin. It will be whether it is a heart cell, or whether it is a lung cell, or maybe a kidney cell, whether it will be a skin cell or a cell that's inside of the body. So each one of these genes have a function together. Together they work to display a certain function or a certain functionality to the body. Okay, so let's just recap. So you have a cell, and inside of the cell you have the nucleus. Inside of the nucleus we have our chromosomes. And a chromosome is basically like a ball of yarn. It's DNA that's rolled up many, many, many times. And if we roll it out, that single strand, we will then call DNA, the entire strand. And parts of the strand have a function to do specific things. So each part will have a function to do a specific thing. And those functions we call genes. Okay, let's move on. So to oversimplify and recap what we learned today is DNA is like the entire book of having all of the information. So it's the entire thing for us to live. While a gene is like a single page from the book that will have specific information on that single page for a particular function. So epigenetic markers, we know now, they sit on top of the genes. So they will, if you think back at this image, so if you have your nucleus with your chromosome and it's rolled out, you have your DNA. We see on specific genes, we have a marker on top of that gene that will either turn it off or turn it on. And so that is what an epigenetic marker is. It's on top of the genes. It is a marker that turns off that gene sequence and on that gene sequence. Now, if you think about it, these markers are very important. If you think with regards to eye color, we don't have one day blue eyes, then the next day brown eyes, then the next day green eyes. Certain markers are determined at conception and they remain on or remain off. For example, I have green eyes, which means the markers for brown eyes and blue eyes on my gene sequence is turned off, whereas my marker for green eyes is turned on. Okay, so this is just very oversimplistic to help you understand. Here's also a website, should you want to go visit it to see how DNA and genes differ. It's a comparison between the two, and it is for beginners. So you'll definitely, if you want to understand more how the genes and the DNA differ. Okay, and that was that for today, is our first session on epigenetic markers and how they work. Okay, I hope you enjoyed learning, and I hope you did learn something with me about DNA and genes and epigenetic markers and how they work. And I hope that you have a lovely day, and I see you next time. Thank you so much for joining me today in this podcast. I hope it enriched your life. Until next time, blessings.